I'll introduce the paper submitted to iMetaOmics. The title is Gene Expression Signatures from Single-Cell Transcriptomics Predict Sjögren's Syndrome. Currently, Sjögren's syndrome diagnosis is challenging due to disease heterogeneity and a lack of specific biomarkers. By reanalyzing single-cell transcriptomic data, we identified 4 key immune cell subsets and built the scRNA-seq model. Its performance was validated across multiple datasets and patient samples, providing a clinically useful tool for early and accurate detection. Highlights. First, decreased naive CD8 positive T cells and Helios FOXP3lo CD4 positive Tregs cells. Second, increased CTLA4 positive CD8 positive inhibitory T cell and TRDC gamma delta T cells. Third, the expression levels of P genes were significantly upregulated in SJS patients. Fourth, developed a validated clinical prediction model for Sjögren's syndrome. Beginning, we reanalyzed PBMC data from SJS patients, capturing over 57,000 high-quality single cells. We identified 11 major immune populations. To ensure high-fidelity annotation, we cross-verified clusters using nebulosa density plots and marker gene heat maps. This refined atlas provides a solid foundation for identifying disease-specific T-cell subsets. After defining major cell types, we established a standardized pipeline to obtain signature genes for key immune subsets. Taking cluster 3, naive CD8 positive T cells as an example, selected genes included: first, cluster-specific genes; second, pathway-specific genes with functional enrichment; and third, differentially expressed genes with high SJS expression in the cluster. The same approach was applied to the other 3 key subsets: cluster 17, cluster 15, and cluster 19. To validate these signatures, we employed a 2-step verification. First, AU-cell scoring confirmed the cell type specificity within our single-cell data. Next, we applied lasso regression to refine the gene sets and performed GSVA on 6 independent public datasets. The significantly higher scores in SJS patients across all cohorts demonstrate the strong robustness of our identified signatures. To validate our findings, we recruited 20 subjects for flow cytometry. Protective subsets cluster 3 and cluster 17 decreased, while inflammatory subsets cluster 15 and cluster 19 increased, matching predictions. qPCR on another 20 new subjects also matched. We validated core markers from cell to mRNA level. We next translated our findings into a clinical tool. By constructing a PPI network and applying code and CytoHubba algorithms, we derived a 12-gene SJS-seq signature, which was validated in 2 independent external datasets. These results demonstrate that SJS-seq enables accurate Sjögren's syndrome prediction across different laboratory settings. We also uncovered changes in their communication. In SJS patients, overall interaction strength is significantly increased, reflecting a pro-inflammatory environment. Naive CD8 positive T cells show marked communication downregulation. Meanwhile, gamma delta T and inhibitory T cells show significantly enhanced communication, identifying them as core nodes mediating immune dysregulation. Summary: our study identifies 4 key immune subsets and establishes the 12-gene SJS-seq model as a reliable diagnostic tool. By providing both a high-performance biomarker and new insights into immune communication, we believe this work offers a promising framework for the precision diagnosis and future therapeutic strategies for SJS. Thank you for your attention.